This is a production of Cornell University.
Yes, thank you so much, Wojtek, for your introduction. Thank you for the organizers to give me an opportunity to share some part of my project with you. Today I will tell you, about the meiotic recombination, how to track and how to answer the question how the meiotic recombination works in maize. But before I will go into the molecular part of it, I will tell you what is a molecular meiotic recombination. It's a process of the genetic material exchange in parent, which are going to the offspring. We have the two homologous chromosomes marked in blue and red. Then in the crossover process, in the meiotic recombination, they are passed to each other, and later they are transferred into the offsprings. And we can use this feature later to create the elite germplasms with the new combination of traits. We can have the gene part responsible for drought resistance, insect resistance, or increase the yield. And simple, if we'll understand how works the landscape and decision process of the meiotic recombination, then we can try to control it and make our own new elite germplasms, and we can accelerate the breeding process, which already take years and cost millions. Meiotic recombination takes place in the process called meiosis, in the meiocytes. The meiosis is subdivided for the many different stages, substages. Starting in the leptotin, here you have the DNA, genomic DNA composed by two homologous chromosomes marked by red and blue. The first step is actually doing the double strand break by the SPO11. Then this double strand break is processed to have the overhangs, single-stranded overhangs in the zygote, and these overhangs start to invade the homologous chromatid and create the intermediate of the crossover intermediate. And this intermediate is going into the non-crossover, which is like gene conversion. The short fragment only is exchanged, or bigger fragment in the crossover type of exchange. The later these crossovers are visible in the diplotin. Under the microscope, you can see how the two chromatids are connected together only on this junction, on this connection of the crossover. And they are resolved in the diakinesis stage. Then our question is how the meiosis is regulated, how the meiotic recombination is regulated. How from 300 to 500 DSBs, high number of these DSBs, at the end, we have only 10 exchanges or to 20 exchanges of the genetic material, the crossover. And why these exchanges take place mostly on the terminus part of the chromosome. We have elevated numbers of the crossovers at the distal part of the chromosomes. However, the central part is limited of it, but there is a lot of the interesting genes which are useful for the breeding. Okay, and how to answer these questions? I was looking on graph, and we found that there is MLH3 protein, which is engaged in the initiation of the recombinant, initiation of the establishment of these intermediates. They are going for the process of these intermediates in the pachytene, and finally, the MLH3 is still engaged in the resolution of the crossover. And the numbers of MLH3 in a process of meiosis is going down through the progression of the prophase 1. Saying that we have the pictures here, microscopic picture, where we have the red dots is MLH3 staining on the zygote stage, where the number is around 30, 40, 50, goes into the lower number in the pachytene, where in the pachytene, I hope you can see, there is this spaghetti stuff. There are chromosomes combined together, there are synapses each other. And the dots is actually where the crossovers take place. And in diakinesis, what I was telling you about this junction, this connection of the homologous chromosomes, these chiasmata sites are marked by the MLH3. And what we can see, there is a selective processing of these crossover intermediates. I mean, what is happening? Why from 50, from the beginning of 40, from the beginning of MLH3, we have at the end only few, which mark exactly the crossovers. But what is MLH3? It's a partner of MLH1, and they together create the complex called mut-L gamma. They interact with the several different meiotic recombination proteins. The downregulation results in the sterility or semi-sterility in many organisms, and has a function in DNA repair and also in the meiotic recombination, what I was telling you already. The method, the tool for tracking this protein, what I use in my studies is chromatin immuno precipitation followed by the next generation sequencing. And here you have the particle of the DNA, which will symbolize here as a recombination intermediate DNA. The red one will be the place where the recombination takes place, and we have the intermediates of it. In the zygote stage, as I said, there's establishment of these intermediates. Then later on the packaging, we have the stabilization, and diakinesis, we have the resolution. But in all these three stages, we have the MLH3 on a place. And when we take the myocytes, the tissue from these particular stages, we are able to pull down this protein. The process looks like this. We have the MLH3 on the particular stage. We cross-link this with the cross-linking agent. We do the fragmentation to have the smaller fragment. We pull down this with the specific antibodies and release the DNA, and we can figure it out where the intermediates were or where the crossovers is. Okay, but before, there was no commercial available antibodies, and create our own recombinant proteins called MLH3N and MLH3C, targeting the specific terminal site of the MLH3. We send this for immunization of rabbit and guinea pig, and we obtain the antibodies. We test these antibodies on a slide. This is a packet in maize. And what we saw, we were very happy because we hope only one will work, but we have the two antibodies working, and they colocalize very well here on the slide. And we were very happy because we have the internal control, saying that if the one antibody will show us something, then okay, maybe it's a noise. But if two on the same side, there's something meaningful. Then another, because this was on the slide under the microscope, but we have done also ChIP and compare the sites of the MLH3C and N, and we got the 80% of the peaks from both overlap with each other in the zygote, 63 in the pachytene, and in diplotin around 80%. And where are these peaks? Where are the intermediates and where are the crossovers?
To remove the
I have to click zoom here.
No, I want to get rid of the
It's high floating meeting controls. High floating meeting controls. It's two down.
to the
Yeah, this one.
okay, brilliant. Now we had someone on the chat box, which it looks very different on the Mac when I do it. So the person on the chat box, could you send me an email to WP45? Then I can check it in my phone.
Okay, thank you, Wojtek, for helping me with this. The MLH3 in the zygote is actually distributed along of the chromosomes. This is a 10 maize chromosomes combined into one, where we have the starting telomere and end of the telomere, and in the middle we have the centromere. And this is a distribution of MLH3 peaks along of the 10 chromosomes combined together in one graph. And what we can see in zygote, we have everywhere. Oh. Yeah, but now I cannot go
Come this
to
up.
the next slide. Oh, wait.
Just look on this slide.
Okay, now should be good. Okay, I'm sorry. And this distribution is very similar for the distribution of double strand breaks, which is very nice because we have something which is looking like something in the zygosine. But later in the pachytene, we have much more peaks on the distal part of the chromosome, on the chromosome ends, which is mimicking the crossover pattern, saying that we see the characteristic typical for the DSBs in younger stages of the meiosis and the crossovers in the later. Simply, we try to assure ourselves we are on the track and we are finding the good things. And later, when we go to the fine scale and we check the MLH3C and N in the different genic or non-genic region and UTRs, which is untranscriptional regions, where the percentages of MLH3 corresponds to the real crossovers mapped by the genetic mapping. And you can see we have like a 10% to 15 where is the control. Here we have the 15 and to 10. But here in the genic region, we have very similar, like a 25% in total for the MLH3 and 25 for the crossovers. Around 50% for the intergenic and 50% for the positive control, and completely different than the negative control. Then how good is MLH3 in detecting the crossovers? And if we will take the 500, the strongest peaks in my ChIP, we are able to detect more than 50% of the crossovers. In compare with the negative control is around 50% in all stages. But I was asking, and where is the other ChIP? Where are the other peaks? And we start to validate the map of where are the hits. And the hits were in the S&P desert, because in traditional genetical mapping, this means how the people are checking where are the crossovers. They simply take the one inbred, B73, and the second inbred, MO17. They know where are the single nucleotide polymorphism or where are the indels. And simply, later they do it the hybrid and they sequence the hybrid. And they know, okay, this part is from the chromosome A. Here we have the S&P from B. And we know here is a crossover. Well, somewhere here. But sometimes we have the crossover, which is out from close by S&P or indel. And we found that more than 40%, more than 45% of the total peaks of MLH3 comes from this desert. Simply, probably, we are able to detect the crossover, but current reference map, what we were using, was too low, the resolution was too low to detect it. And using, maybe we can overcome now this limitation. We don't have to use the S&Ps. And we create the first time ever the crossover inbred line. Where we don't use any hybrid, and it's very robust and efficient. We have the different window sites here, and we compare this with the NAM population, which is nested association mapping created by the B73 crossed with the multiple inbred lines. And we compared this sites of the crossovers with our MLH3, and we got more than 65%. But still, we can have some limitation because of the resolution of our reference crossover map. In the pachytene, we can also see there is more elevated peaks on the distal part of the chromosome. If we had finally the peaks from the B73, we start to analyze where are these peaks, and we found the motifs associated with this MLH3 binding site, and we found like a polythymidine sites or very, very conserved, high specific sequence or palindromic sequence. And when we try to check and validate where are these palindromic and where are these highly specific sequence, we found that they are coming from the 180 repeats of knobs, and knobs are very heavily methylated and very condensed heterochromatin structures. You can see here on the picture with the MLH3 staining inside. And this was surprising for us because the crossovers from the previous studies, we know the crossovers doesn't like the methylation and do not like the heterochromatin. However, 25% of the total peaks was coming from the knob sites. Yeah, but not all of them. When you can see on the chromosome 8, we have here the map of the repeats, and the red is marking where are the repeats, and the blue where we have the hits from MLH3. We are still on the way to analyze this. There is also the two features, which one I was mentioning. The methylation is associated with the crossover, and the second one is heterochromatin, which is also associated with the nucleosome position, simple how many nucleosomes is there and how dense is chromatin there. And we found that in the zygotin and pakitin, we have slight transition in the peak. Here we have the medial binding peak or binding site of the MLH3, which is narrower than the pakitin, saying that we have some kind of the selection pressure to more relaxed chromatin, less saturated of the nucleosomes on this position. When we go to the methylation studies, we see we have three different types of the methylation in plants, which is predominantly CG and CHG, where the H is everything except G. The dotted line is reference of the genome. The CG and CHG is here, where they downregulated in the zygotin and the binding site of MLH3. The CHH is only like a 2-3% of the total methylation. It's a little bit elevated, but we see the transition and some preference for the sites which present the deep of methylation types in the CG and CHG, and elevated signal from CHH, which is quite interesting. We have some kind of the selective pressure for these sites, which shows the typical characteristics in these two pictures. Then how meiosis is regulated. We have from 300 to 500 DSBs. Then later on, we have the resection map, which we are also working about. Then we have the zygotin map of MLH3, where we have the 3050. And then we have the transition for the specific sequence. Also for the knobs, we have the nucleosome depletion factor, which is also preference for being a crossover, reduction in CG and CHG, and increase of the CHH. And finally, at the end, we have the crossover. Well, to sum up this everything, MLH3 detects the crossover site with the high accuracy in hybrid, and it's possible to use this in the inbred. In the pure lines, this means we don't have done artificial crosses, but we can do it in the pure line. This really reveals several types of the binding associated motifs. It's much cheaper. The method is much cheaper, more accurate. We can do it on the nucleotide resolution scale. It's much robust. We can survey a million of crossovers instead of the thousands, and much cheaper. You don't have to build a population and sequence a lot. And the cytological and ChIP data show us that the MLH3 has a role in the crossover, non-crossover decision, and also the heterochromatin, the chromatin features and sequence features like knob and nucleosome density may push for the selection pressure for the crossover. At the end, I would like to thank you so much for Minghui Wang, who is doing this all magic with the bioinformatic data. Kasia Zelkowska for helping me with the wet bench work. Wojtek Zelkowski for his experience and sharing of the knowledge. Thank you, NSF, for founding, Cornell for hosting, and thank you all for your attention.
very nice talk. So could you just go back to the slides showing the distribution of MLH3 binding target sequence at 3 different crossover stage? Yeah, so this one. So you can see the zygogen, so distribution of the MLH3 binding is just a little different from other 2 stages. So this one, other 2 stages, basically they have more, binding target at the distal region of the chromosome. But this one more like somewhere in the middle. So could you just explain a little bit about why this happened? Or
Mateusz, can you repeat the question?
The question is why we have the difference in distribution in zygotin and pachycine, and whether I'm able to explain it. The zygotin peaks, which is along of the chromosome, is mimicking the DSBs, and I think they are simple. They are combining in the processing of the early stage of the meiotic recombination, saying that it's still sitting here on the DS. This is data from RAD51, which is DSB marker, and simply we can see this along of the chromosome. This is also related with some histone marks, which are preferable for RAD51. And what I can say is that first we have plenty of the DSBs; they are everywhere. But later on, we have the transition into the sites, which shows these features. What I was showing later, this nucleosome occupancy and methylation state. I don't know whether do I explain.
Okay, that should be fine. So this distribution pattern is similar to different, like B73 or B317, or just specific to certain genotype, certain background.
We tested only B73, but I would say this is common for all different IMREDs.
Okay.
For the crossover distribution in maize, there is always U-shape. We have more on the distal part and less in the central region or intersectional region.
Thanks.
I should know this, but I don't. What's the cause of the double strand breaks?
What is the cause?
The double strand breaks.
Yeah, this is what is the cause of the double strand break? Is that because if you would like to have the exchange of the genetic material and finally to have some heterozygosity, simple evolution itself, then you need to do the first step to cut the DNA and somehow physically allow this process to happen. That's why we cut one, and to have the invasion later on and exchanges to have the crossover.
Is it an enzymatic action or is it physical?
It is enzymatic, mediated by the SPO11 protein.
They programmed, so they're not accidental. No more questions? Let's thank Mateusz and.
This has been a production of Cornell University, on the web at cornell.edu.